Medical School Histology, Images of the Salivary Gland. Today you'll be able to download an image to manipulate on your computer of the salivary gland. You'll be able to see the different cells that are there, the different types of ducts that are there, and to examine overall structure of the salivary gland. Medical School Histology, Images of the Salivary Gland. Hi, I'm Larry Johnson. I'm a professor at Texas A&M University. Please consider subscribing to my VIBS Histology YouTube channel. Today we want to look at salivary gland, and you'll be able to observe the salivary gland, interact with it, manipulate it, change the magnification, make measurements on the cells if you go to this website, as we'll see there. And you can locate those websites at this website. This website gives you this URL to be able to do that. And also the PowerPoint slides can be seen on YouTube where it says to see PowerPoint slides. So remember, we were talking about epithelium. There are different shapes of epithelium, but a characteristic of epithelium is they have junctions between adjacent cells. And those junctions allow cells to be attached to one another to form a sheet and to make a cavity. And here we see in the kidney where the cells have made a lumen or opening, and through there that fluid can go through. And we're going to see that in the salivary gland today. So the functional unit of the salivary gland is an acnes, which is a group of cells, kind of like a grape hull of cells with a lumen or opening in the very center. Now, some of the salivary gland secretions are mucus type, and here you can see mucus type. Usually the mucus type has a flattened nucleus at the bottom and then a flocculent material in through here to the lumen. The other type is serous, which is more proteinaceous, and those, the nuclei, are usually not located all the way at the base, and usually they're spherical nuclei, spherical versus flattened. Off the base, on the base is the two type that we can see. Now in our slides today we will see this is the serous type, serous secretions and a more spherical cell, and then here we see the mucus type. This is the mucus secretions and maybe a more flattened cell at the base. Now, sometimes you have mucus and serous all together, and we'll call this a serous demilume, where the serous secretions actually go through these intercellular canaliculi to go into the lumen. So regardless of where they are, they ultimately find the lumen. Also note that you had myoepithelium, which is a type of epithelium that surrounds the secretory portion and squeezes out the secretions. And we'll see those today as the little flattened cells around the acnes. If this is the acnes, the functional unit, then these guys here, here and here, are the myoepithelia cells that squeezes out the secretions. So here we see a demilume, as I mentioned, where you have the serous on the outside of the mucus, and then you can see where junctional complexes are between cells and make a lumen. And this is the intercalated duct, which is simple cuboidal epithelium. And then you come to the striated duct, which are more columnar type of cells. So we got the secretion, serous, and mucus, intercalated, and striated duct. So we can see the order of which you have. You have the acnes is a functional unit, intercalated duct transports the fluid to the striated duct. The striated duct modifies the fluid, as we can talk about next, and then the excretory duct takes it outside. So in the secretory portion, especially the serous, which are proteinaceous granules through here that be discharged, has a lot of rough endoplasmic reticulum for protein production. Intercalated duct has less so, and then the striated duct is unique, and it's striated because the mitochondria are lined up, and they're lined up because the plasma membrane has increased its surface area by actually projecting into the bottom of the cell. So this is a striated duct. So if we look at those today, we'll see these. This is the acnus. This is an acinar cell right here, salivary acinar cell. There's the lumen. The lumen is really not that big, it's quite smaller. And then you have intercalated duct, kind of cuboidal cells in through there. You can see the secretions of the serous secretions in through there, but that's the duct. And then here's a striated duct, and you can see Loma Hoa has striations. And then there's excretory duct. So we can see those again. The acnus, and then the intercalated duct, the striated duct, and the excretory duct. Usually excretory ducts start out as simple cuboidal, and then they go to simple columnar, and then they go stratified columnar, as you see here. There are some cells at the base, but these are columnar cells at the top. Some of them are just regular columnar cells of a duct. Some of them are actually goblet cells as well. And so, as I mentioned before, with epithelium in order to form a sheet, whether you're in the acnus, where you're in the intercalated duct, or the striated duct, or excretory duct, you have to have these junctional complexes. So you have the zona occludens, which prevent things from going through, from losing your saliva from the duct, and then you have these other junctions, and we'll see these junctions here. Remember, we talked about the terminal bars in the intestine, but it's really a belt that goes all the way around the cells, as we talked about before. Here you can see those, and you can see that the mitochondria are lined up as you get the infolding of the plasma membrane, and it's the mitochondria being lined up that you see as the striations. Here again we can see the striations, and this is an electron micrograph showing you the mitochondria and the infolding of the plasma membrane that goes in through there, which increases the surface area of the base. And again, we can see that actually these are mitochondria that are lined up that gives the striated appearance. Now, whenever the body produces fluid, it produces it in an isotonic state. That is 300 milliosmoles is what it is. However, in saliva, the striated ducts pump sodium out. That's why you have the infolding and the increase of surface area to pump the sodium out to make a hypotonic solution. Isotonic goes to hypotonic and is brought about by the striated duct, which pumps out the sodium. Adds some potassium, but it pumps out the sodium, which reduces the osmotic pressure from 300 to about 180 or so. Now, so we can look at this specimen. We actually go here and go to the website where this specimen is, and we will see the specimen. And here we come, and here we can see the specimen. This is a gross specimen. There's connective tissue around it. As you can see, it's really embedded in this, not in a capsule per se. And then here is going to be the excretory ducts going out. There's more excretory ducts in through there in the connective tissue, and these are the different lobes. So the interlobular and intralobular ducts that we see. So if we just look at one of these, we can see here's a duct that we see. This is a duct. You see the columnar epithelial cells lining that. That's in contrast to this vein. Yeah, it looks like a vein with some smooth muscle, but you see endothelial cells on the surface. So this is the duct cell, and then here another. This is this little artery. You can see the internal elastic lamina in through there. Endothelial cells, smooth muscle cells, but these are columnar cells that you see there. And here you can see the striated duct in through there, and these are the acini. So these are the acini. There's one right here. There's another one right there. You can see the lumen of these, and these are the serous ones. Serous are pronaceous, as opposed to the mucous ones. The mucous ones, remember, have the flattened nucleus at the base as opposed to spherical nuclei. And so you have a series of acini that kind of run in together. Here's one that has both mucus and serous in there. And so this is a blood vessel right there. You can see the endothelial cells, a capillary. So coming from the acini, you're going to have the intercalated duct. So here's the intercalated duct right here. One that you can see, simple cuboidal epithelium with a lumen in through there. In contrast, from there it runs into the striated duct. And here we can see a striated duct. There's another one here and here. So 1, 2, 3 striated ducts. And you can see the striations, which is where the mitochondria are lined up. And right around there we see the myoepithelium. The myoepithelium that squeezes the secretions out. Let's see, let me see, make sure that's right. This looks like more myoepithelium right in here that surrounds the ascens. Maybe that's fibroblasts in through there. So the myoepithelium you would expect to find around the ascens itself like this. So myoepithelium, remember, squeezes the cells out of ascens. This would be like a fibroblast right in the loose connective tissue. This would be more like the myoepithelial cell nucleus that we would see. Striated ducts, and this is an intercalated duct that's cut longitudinally. So it coming out of the ascens. This is a mucus type, serous type coming out through there. And so from there we can go to the lower mag, and we can go to the ducts. Here it is an artery going in through there. And we can see goblet cells as well as goblet cells, regular cells, and we can see these cells at the bottom. So this is stratified columnar epithelium. There's an artifact right there. Don't worry about that. But here we can have this actually nerve in through there. This is nervous tissue. There's the perineurium right around there. There's blood vessels. Here's probably a lymphatic. Probably a lymphatic, very thin endothelial cells going in through there, as opposed to this vein. So vein right there, and here's another nerve. So you can see nerve, I can see blood vessels in the nerve. There's a perineurium. These are swan cells surrounding each of these axons. You can see the axons here, which would have a swan cell cytoplasm around there. And someplace in here there's some ganglion cells. I'm not sure I'll be able to see that. But basically what we're supposed to see is that we have the serous and mucus, serous secretory products. You have a striated duct. You have intercalated duct. These are fibroblasts right in through there for sure. And then you have the excretory duct. So these are the striated ducts that are inside the lobe of it. Okay, so we go back, and then, so in summary, in the salivary gland we have the ascens, we have the intercalated duct, we have the striated duct, and we have the excretory duct. And the reason we have striated duct, the unique part about salivary glands, is that we have striated ducts. We don't have striated ducts in the pancreas, but we do in the salivary gland. Why? Because we need a hypotonic saliva. So this ends the medical school histology, images of the salivary gland. Thank you.